So, super cool guy. So for our course, let me go ahead and go into it. So just remember that everyone took the test tonight, which is awesome. So I was able to turn on the results for your test. But to review your results, you have to have the lockdown browser, which most of you guys are downloading anyways because we want to be able to test in class. So you just do like you did in the computer lab. You make sure everything else is closed on your computer. You hit lockdown browser. You log on to Canvas and you click on the test, and then you scroll down and it says review results. And you click on that, and you will actually see what ones you got right or wrong, what the correct answers are. And what I really want you guys to focus on too is in the feedback section, what objective was linked with that question. Now, for some reason with the matchings, the feedback wasn't working when I set up these tests. But you guys can pretty much tell too, like by the question, which objective that went with. So, you know, you have to do a little bit of work, but not too much. I don't think I had too many matching on this test even. So, and as I said, you know, the average was really, really good, and nobody's in major danger zone. So that's really good news. Where did I want to go? Oh, modules. Okay. So one of the things that you guys need to do before the end of the course is the case studies. So I actually have instructions. I don't know how many people actually read them. But within these instructions, the Listeria case study, there's 6 case studies. The Listeria one is the only one I basically don't give you the answers to. I want you to read over the case study. I've even put photocopies of the book there too, so you know exactly where you would need to read. And then there's PowerPoints for all of them, and even recorded lectures for all of them except for Listeria. So Listeria you got to kind of do on your own. So what I always suggest to my students is, you know, print out the PowerPoints for each one of them, print out the case study, read over the case study, read over the textbook stuff, see how much you can figure out on your own. Then watch the lectures with the PowerPoint, right, where you could take notes like you kind of do for regular lecture class. All of those notes that you take, all that stuff you compile, you can use to take the test for the case studies, right? And that one you don't need the lockdown browser. You can open it right in Canvas. But once you open it, you cannot open it again. It's not timed or anything like that. You can take 4 hours to do the test if you want, right? What you need, Charlie?
I'm sorry to interrupt.
It's okay.
The volume on my system isn't working. Do you know why that might be?
I know several of the tricks. Give me 1 second and I'll come try and help you.
Okay.
For the Listeria one, so if you just go to next, I have kind of even pared down the questions from the case study. These are the ones I really want you to be able to answer. So it's written in that first document. And then I don't understand why it won't go to the next screen. At first I liked Canvas and now I'm not so much in love with it. But so there's the questions I want you to be able to answer. There's the actual case study itself. Here's from a book other than yours that really summarizes it pretty good. And then once you think you figured out the questions, go ahead and do this case study assignment. It's a quiz in Canvas. And when you get done, it'll tell you the answers. So that way, when you go to take the test on all the case studies, right, you know the correct answers to the case study on Listeria. Make sense? This is a 20-point assignment, and then the test is 100, and it is already published. And the due date is at the end, July 28th. Actually, that's probably too late, right? I probably need to change that one. We need to be done by the 22nd, I think. What's the last day of classes? Yeah. So, oh no, that's July. Last day is the 20th. So this is one of those things you guys could get knocked out when you don't have, since you don't have to come to class next week. But you still need to work on Module 2 stuff. right? Because I'm not going to lecture on all of it when I get back. I'm going to do kind of an abbreviated thing, probably even an activity with you guys, see how much you remember. All right, let me pause. Questions regarding case studies and what you need to do for that.
So we just have to read them and then answer the questions, or just read them and keep things in?
Right. So your best bet for Listeria, you've got to read it, answer the questions, and do the quiz on it. For the rest of them, right, you can read it, you can watch the video, you can download the PowerPoint, you can write down all the answers, organize all your notes, and then open the test, have your notes in front of you, and take the test. Yeah.
But for Listeria, you have to.
Yeah, for Listeria, you've got to try and figure out the answers on your own, and then take the quiz, which will tell you the answers when you get done, right, but not before. You got to do a little bit of work, not too terribly much. And I didn't pick Listeria because of what happened with Bluebell. But it fit in well. Anything I talk about in this class always ends up happening.
I'm still scared to eat. Eat what?
Yeah, but Listeria isn't going to hurt most people. It's people who are immunocompromised, people who are pregnant, which, you know, when you're pregnant, it's usually the only thing you want to eat, and and elderly, is that bacteria really can be quite damaging, as you'll read about in the case study. All right, so moving on to finishing up where we left off last time, which was viruses. So we made our cute little virus models, right? We had fun with that. And so we'll finish up viruses, and we're going to start on genetics. And genetics is usually kind of the tough stuff, which is why I usually deal with it pretty first. And then the last two chapters, the last two topics is growth, and then I continue talking about growth, but then I add in control. So another thing that you guys have to do is that next discussion board on chemicals used to clean. So if you guys could get that done before I come back, that would be great, because then we can talk about it when I come back. So I open up viruses, let me open up genetics. And that's a really fun discussion board. Students really like that because it's interactive and you learn stuff that applies to your everyday life. So whenever you can relate to it on that level, right, it's always good. And I know probably, and I didn't even look for that. But remember, Connect assignments I don't have linked up with Canvas, but just to see where you guys were at before the test, you'll notice that I put a score in for the first module in Connect, what you had completed. That's not a permanent score, right? That's going to change. I'll check it again later at midterm to do midterm grading. And I'll check it again before the final, before each test, before the final, and then after the final, when that's it, I get to turn in grades. Your maximum score, right? And again, it's the best score on any of your Connect assignments that counts. So you could potentially get 140 points out of 140 because you can do the assignments over and over again until the end, until I say, it's over, y'all. That's it. Okay, and then there's some bonus ones too, which one of them relates to this, which I know you guys were studying for the test, so nobody watched them. So that's another thing to do. Especially if, seeing stuff in motion is helpful to you guys. I'm not going to spend the time right now because we're short on time to play those. But, to actually see stuff in motion for some people, that really helps, right? Which is why I assigned them as bonus instead of making them required, because some people don't like them, right? Some people are like, this is a complete waste of my time. It's driving me crazy. So to each their own, right? So where we stopped last time is right about here, right? We were talking about influenza, and see, remember I told you guys sometimes I have the notes in the notes section, right? That tells you the answers basically. So it's hemagglutinin, right? That's important for the attachment, but it's that neuraminidase that's enzymatic. When it enters into the cell, it shuts down that cell's receptors that the hemagglutinin binds to. So they're immune to what's called superinfection, where only one virus gets into a cell at a time, the influenza virus. They're very greedy. But this also helps with the spread because only one virus per cell, so all the other guys have got to find another cell to get into. Superinfection? Superinfection is when more than one virus gets into a cell at a time. And this is one of the ways that some viruses very quickly mutate, though. Because if more than one virus gets into a cell at the same time, they may mix up their genetic information. And then you get a new virus out of it. And that's happened numerous times where we've had these really big jumps and changes in viruses. I think they believe that happened with the swine flu. Because that virus is very similar to two other viruses. So it's almost like viruses can mate too. Oh my Lord Jesus. It's just all about it. So the other interesting thing, and remember, we're kind of concentrating a little bit on viruses that infect bacteria because this is a microbiology class. But similar things happen in us too with certain viral infections. So there are two different, what are sometimes referred to as life cycles or reproductive tracts that some viruses can take. One is referred to as the lytic, the other is the lysogenic. And so some terminology that relates to these is temperate phage, and phage, remember, is the name given to viruses that infect bacteria. The full name is usually bacterial phage. Virulent phage, lysogeny, and induction are important terms as we talk about these different types of viral infection. So what a temperate phage is are phages that have two reproductive options. They can reproduce lytically, which means they keep lysing, right? Lytic means lyse. They keep lysing out of cells and going into new cells, reproducing and lysing out of them. So that's referred to as a virulent phage because it's actively reproducing right inside the cells, busting out, infecting new cells, and it keeps doing this over and over again. Or they can choose to remain within the host cell without destroying it. In that case, this condition is referred to as lysogeny, where they actually incorporate their DNA into the bacterial cell's DNA. So when that bacteria divides, it actually copies the virus's DNA as well. So here is it diagrammically. So temperate phages can go lytic cycle, where they're going to reproduce inside the cell, lyse out, and infect more cells. And they can just keep going around and around like this, over and over again. Or their DNA can incorporate into the bacteria's DNA. When it's in that state, it's referred to as a prophage, right? Phage again meaning the virus. Pro meaning it could become a nasty virus again. As I said, when the bacterial cells copy, they copy the viral DNA as well, the prophage. And this can go on and on and on, and so all these bacteria could be, you could say, infected, right, with this virus. At any point, due to several different triggers, it can be induced where the prophage pops out of the chromosome. This is called induction, and it could enter into instead the lytic cycle. And then, of course, at any point, it can go from lytic back to lysogenic, where it hides in the chromosome. So prophage, as I said, when it's integrated, when we have lysogeny, when it's actually integrated into the DNA of the bacteria. lysogens or lysogenic bacteria, that's referring to the bacteria that's infected. The name to the phage, the viruses that infect, are temperate. And so therefore they're able to establish this, what we call lysogeny, where they're basically hiding out. They too are immune to superinfection, so one virus per cell, right? It usually doesn't allow another virus to get in, because that would disrupt the DNA even more in the bacteria and likely could cause the death of that bacteria, which means it would also cause the destruction of the original virus. And then, as I said, they're under certain conditions, like in that diagram, UV light, which can damage DNA, which would cause repair mechanisms, which would stimulate the cell to change and that prophage pop off and go into the lytic cycle. So remember, these are pieces of DNA which code for stuff. They can be translated or transcribed first into RNA and then translated into proteins. And they found that actually there's a modification in Salmonella to their lipopolysaccharide layer, right, that outer membrane layer, that's actually because they're infected by a prophage, by a virus. Diphtheria. The reason why we fear Corynebacterium diphtheria is that long ago, this bacteria got infected by a temperate phage and began this lysogenic relationship where you have this prophage. The bad news is that viral DNA encodes for the toxin that this bacteria produces. So not all Corynebacteria are pathogenic, but the ones that have this prophage, which is most of the Corynebacterium out there, produce the toxin that causes the disease that we know as diphtheria. And we actually vaccinate people against the toxin, not really the bacteria. It isn't really necessarily the bad news in this situation, it's the toxin it produces. It'll kill the epithelial cells lining the reproductive tract and will literally cause the cells to die, and you have this dead membrane that can fall in the throat and block the throat and you suffocate to death. It's referred to as a pseudomembrane. So the Iditarod, I don't know if I'm saying that right, the race in Alaska of the sled dogs is actually a reenactment of a historical event that happened in Alaska where they get locked in when the snows come in some of the areas in Alaska. And they had an outbreak of diphtheria. And so they rushed, and I can't remember, I think it was the anti-serum. They were rushing back to the community that had this outbreak of diphtheria. It was too cold, planes couldn't fly, even though there were planes back then. So the sled dogs relay raced the anti-serum back to that community and saved them. So pretty cool stuff. So for eukaryotic cells, there are cytopathic changes that can happen when the virus infects a cell. So we already know, right, we live in an age where, TV, we had an interesting lunch conversation today. You gotta love TV. There was an erectile dysfunction commercial that came on TV last night. And my husband and my son were watching TV and eating dinner. Dad, what's that? I was like, oh, how much did you tell him? I don't know if I want to know the fun we have to look forward to. Oh, boy. Right, so on that same lines of sex, right, and how all this stuff happens, you can transmit things to other people, like viruses. And there's one group of viruses that we worry about being transmitted sexually, and those are the papillomaviruses, because several of them have been linked to cervical cancer for women, because we're the only ones who have cervix right now, unless they decide they want to try and change that. I don't think they do. And unfortunately, you can't look at her whenever you want, right, ladies? She's kind of up in there. So you have to go every year, right, like clockwork, and go get it checked out by the doctor and make sure everything's kosher down there. There's also a vaccine. There's a lot of controversy about it. I haven't really investigated it. I'm too old to get it. I don't need it. I don't have too many worries with that. Just go get my pap smear done every year, right? But we know that those viruses, right, that can cause warts can infect the cervix. And, of course, one of the main ways they get there is sexual contact, right? So males can carry it, but it usually doesn't cause serious problems for them. But that's also why they're trying to get guys vaccinated, because that's typically how it gets spread, right? That's how we spread these things to one another. So those particular, what a wart actually is your cells deformed by the virus. Right? So those are actually your cells that look all strange like that because of the viral infection. So even if you get warts, say, on your hands or your feet, which is the worst place, the bottom of your foot, right? Because then it gets pushed inward and you've got to get it surgically removed if you don't know what it is, like I did when I was in high school, because we were required to take swimming. I wore my shower shoes, but it didn't matter. Right? And when you run track and you miss a month of track because of a plantar's wart, you're not happy. But when you're an adult and you get one from the gym, you recognize it. And you go buy the stuff over the counter at the drugstore and you put it on the bottom of your foot so you don't have to have it surgically removed because it grew halfway into your foot. Right? But what it is again, deformed cells. Right? So you've got to get all the cells, because guess what? Not just the deformed ones have it in it, the ones next to it do. So until you get rid of all the cells that have the virus, the wart just keeps coming back. Make sense? Right? So that's why they like really scrape it or really freeze it and scrape it, right? Depending. The good news is most of them will pretty much go away on their own. I had ones all over my hands for playing in the dirt when I was a kid. They call them worry warts. When my mom went into the hospital, had my little sister, guess what? They all went away. She used to put that tincture medicine on them. But guess what? I quit playing in the dirt because then I had a little baby sister to play with and they went away. Then I had to go to the neighbor's or friend's house when my mom was in the hospital having the baby. So, it's environmental. Little tiny little scratches on your hands or your feet is how it gets in, right? And moist environments are what really harbor these types of viruses that cause warts. And then there's the whole sexual problem too. So the bad news is that they deform our cells. What's cancer? Yeah, abnormal cells. It's basically your cells behaving badly. And there's all different types of cancers because guess what? You have all different types of cells. Right? My husband is convinced there's a cure for cancer and they're holding back. He's not a scientist. I love him. Like, honey, there's lots of different types of cancer. The easiest way to explain it is your cells behaving badly, right? Not doing what they're supposed to do anymore and doing things they're not supposed to do, right? But you've got, I don't even know how many different types of cells, and it really depends on, what cell is doing this, right? And even kept convincing his buddies next to him to do what he's doing, right? That's the bad news with that. Is they're not, one, doing what they're supposed to, and they may even be doing things they're not supposed to, right? That's cancer in a nutshell. Problem is, again, what cell is this, and where is it, right? And how crucial is it to life on this earth? So viruses, unfortunately, can screw with the system inside the cell, right? Hence why we could get to that state where we call it cancer, cells behaving really badly. Still with me? Okay, so warts are, a really good example of deformity, right? Cytopathic effects, right? These changes in our cells that makes them abnormal. So in addition to just multiplying and busting out of our cells and infecting other cells, they can kind of stay inside our cells and cause these abnormalities that we see that can even lead to cancer, right? So the worst case scenario is a transformed into a malignant cancer cell. Other infections, right, slowly release the viruses. Can anyone think of an infection where it's kind of slow progressing and it's a viral infection? It's what we refer to as a chronic. You're sick for a really long time, and it usually takes a while to understand what's wrong. Respiratory, sometimes. Hepatitis. Most of the hepatitises, right, are relatively slow progressing viral infections. So that they go under the radar for a little while for some people because you're really not dealing with the effects. Unlike acute infections, right, where it's rapid, there's no question. The flu, right, when you get the flu, you're like, holy cow, right? The good news about them is they're usually pretty rapid. I mean, they're busting out of your cells left and right. But usually, hopefully, your immune system gets a handle on it relatively rapidly as well, and you recover relatively quickly. The really bad news is some infections, right? You get infected, and then it does like lysogeny, it hides out in your cells. And then every once in a while it goes, ha, let's party, let's bust out, right? Can anyone think of one that does that? So you get the infection, right? You're fine for a while, then all of a sudden, eruption again. Herpes. Yep, herpes viruses. The cold sores that you get are herpes, right? And again, when they're erupting, when you have the sore, that's when they're busting out of your cells. But when that cold sore goes away, the herpes did not. It's just gotten quieted by your immune system. Your immune system has kept it in check. It's hiding in those epithelial cells. So guess when you see cold sores? What time of the semester do I see them? Finals. Because you don't sleep right, you don't eat right, right? You're stressed. This stresses your immune system. It doesn't allow it to work under optimum conditions. So it can't keep the bad guys in check, so they erupt, right? And you can get herpes either place, depending on what you do. And there's type 1 and type 2. Type 1 is typically the cold sores people get around their mouths, right? Type 2 is typically genital herpes, right, which tends to erupt more often. I love those commercials on TV. They're like, oh, I take such and such drug, and I haven't gotten herpes from my partner. And I always yell, yet! Time, probability's going to get you, girl or boy. Right? If your partner's got it, you're going to end up with it eventually. Right? What's the good news about the drugs is they don't have eruptions as often. It's not as painful for them. Right? And obviously when they're having an eruption, that you're more likely to spread the virus because it's popping out of the cells. But again, depending on what you do, those cells in that area are infected. If they get transferred to you, you can get the infection. Make sense? All right. There's one also that we get vaccinated against now. In 1995, they came up with the vaccine against this virus that can reactivate, and it's called a different infection when it reactivates. Yeah, chickenpox. So when I was a kid, your parents purposely exposed you to other people who had chickenpox before you went to school, so you'd get chickenpox before you went to school. Apparently, I was very good at playing that particular way. It's respiratorily transmitted when you have chickenpox, but some people even go to extremes and let little kids share lollipops to make sure they get the infection before they go to school, right? Now, my son will never have chickenpox. He's been vaccinated, right? No more chickenpox parties, thanks to the vaccine. My little sister apparently played very clean. She didn't get chickenpox until she was in high school. Missed 2 weeks of school. Plus, it tends to be worse when you're older. Although I'll tell you, I was 4 and I still remember. Those suckers were itchy. And Mom taped mittens on my hands. Duct taped mittens on my hands to get me to stop scratching. I still have scars from the ones I picked off. So I'm very grateful for that vaccine, that I didn't have to go through my son going through that. But what do we have to worry about? Because basically all of us have this virus. Either you experienced it as a child or unfortunately as a teenager, and God knows you definitely don't want to have it when you're pregnant because it can cross the placenta, right? That's the fear with that. So we've either been vaccinated and probably had it nowadays, right? So we've all got this virus hiding in our sensory nerve ganglion. Ooh, that's the crappy part, because when this guy erupts, it's busting out of sensory nerves. Sensory nerves are the ones that go, ow, 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 ow. Oh yeah, they do ow, ow, ow very loud, apparently, when it re-erupts. Right? So guess when that happens to people? Stressed. Right? So if you're a younger person, you're not in your 70s or 80s, you're in your 40s or 50s, right? You are living a very stressful life and you need to chill it if you get shingles at that age, right? There's something going on, or you've got some other autoimmune issues, right, going on. That's not a good sign. And then, of course, if you're a professor here, we walk around and tell everybody you have herpes because it is a herpes virus. It's in that same family, right? And that's why it does what it does. It hides in your DNA, right? And then it'll bust out when your immune system isn't watching and keeping it under control. It's also why you'll see it'll run along the back, or for some people, they even will get it on their face or along their chest, right? Because it follows that nerve ganglion that it's erupting out of. And that's why it's so painful. So our lab prep person right now, her husband he's a contractor, and he just had it. But he's in his 50s, but still, that's too young to probably be having it. And she diagnosed it, and he waited like 3 days before he went to the doctor. And he wonders why he was in so much pain. He's like, if we would have got antiviral drugs sooner and nerve pain medicine sooner, you wouldn't be suffering right now, and I would want to kill you. They don't listen.
So is shingles maybe transmitted the same way?
Good question. So can shingles be transmitted the same way as chickenpox? Chickenpox is systemic, and it's transmitted respiratorily. Thankfully, the reactivation is not. It is to that area. So if you've never had chickenpox, because this happened to my mother-in-law, she had breast cancer and was going through chemo, and she got shingles. And I was pregnant, and she was freaking out about me coming over. I was like, don't worry, I've had chickenpox, for one. And two, you're not respiratorily transmitting it when you have shingles. So I'd have to come in contact with the pustules, right, with the actual virus in the wounds to actually get the infection from her. So they're not quite as contagious, I guess you could say, when you have a reactivation when you have shingles. The good news, too, is because of the chickenpox vaccine, we can also vaccinate you again. Even if you've had chickenpox, you can get a booster of the chickenpox vaccine. That's what the shingles vaccine is. And again, this is telling your immune system, hey, 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 this is what it looks like. It's there. Keep that under check, right? It's a little reminder to your immune system. So they recommend that for people in their 60s, right? Because it's, if you're like me and you got it when you were 4, that was 61 years ago. If you're 65 years old, right? Your immune system may have forgotten about that little guy on board, and you don't want that to happen. Make sense? Okay, so what's the bad news, right? These viruses inside of our cells can inhibit our own DNA and RNA and protein synthesis, right? This can lead to cancers and all kinds of problems. It can even damage the lysosomes. Remember, those are digestive enzymes, hydrolytic enzymes that we purposely keep in membranes, right? Because we don't want them busting out. Well, they can disrupt those membranes and release those enzymes and basically kill a cell and destroy themselves when they do it. The good news is we're multicellular organisms. We can spare a few cells, right? Few thousand, we can make it, depending on which ones you're talking about, though, right? In the brain, don't really want to spare any of those, but you know, if we had to, we could. Maybe still survive. But they can alter the membrane, and this is a lot of times, like when you have rashes, a lot of them that are viral especially, it's actually, the rash is your immune system attacking the virus-infected cells. So remember, they use our membrane to make their envelope. So they're sticking their little viral proteins in our membrane, and our immune system comes by and goes, duck, duck, oh, goose, that one's not mine. That's not my protein. That's not good. That cell's not good. That cell's behaving badly. It must go, right? It attacks and kills that cell. And that inflammation that we're going to talk about later is your immune system responding. Those white blood cells going to that area and killing the cells that are infected and trying to stop the spread of that infection. It can cause, you know, our cells do weird things like fuse together, even though they're not supposed to do that. Of course, something that's not us building up inside of our cells can be toxic and actually lead to the death of the cell. And then there's those inclusion bodies that I mentioned before, and especially for rabies. There's a particular structure to these viral crystallizations that happen for rabies in the Purkinje cells in the brain. So they can look for that and diagnose that way. It can disrupt the chromosome, and again, the worst case scenario, we have malignant cells. So those are those Niagara bodies that I talked about earlier, and that's for rabies. So when they, you know, kill an autopsy an animal, they're looking at slides they make from the brain tissue, and they're looking for these structures, these inclusion bodies that happen with rabies infection. And if it's a raccoon, they usually find it in Louisiana. The #1 carriers of rabies. Leave them alone. So that's it for viruses. And they're a good prelude to genetics, right? Because we talked about their DNA and their RNA and that kind of stuff. So it kind of warms us up for genetics.